everyone, in this video we're going to talk about midterm review part 3, mixtures and pH and macromolecules as well as enzymes. So we're going to start with mixture and pH, the difference between a mixture, solution, and a suspension. So a mixture is basically you take different material and you mix it up. It could be any size of a material, it could be any amount of material, it could be many different types of material. So you put it into a mixture. So water, for example, salt water, for example, in the ocean, there's a lot of things in there, right? So that's a mixture. However, in comparison, a solution has to be a homogeneous mixture, meaning that everything has to be mixed up evenly and that the different parts of this mixture doesn't separate on its own very easily. So for example, if you just take a cup of water, you mix in some salt, you stir it around, you can't see the salt anymore, but you can taste the salt, so you know the salt is in there. But the salt and water doesn't separate on its own if you close the cap of a bottle, for example. Salt water in a bottle, close the cap, right? So a solution is this even mixture where it doesn't separate the different parts of the mixture on its own. A solute is something that you dissolve into the solution, and the solvent is what's being used to dissolve the solute. So in terms of salt and water, the salt will be the solute and the water will be the solvent. Normally, you would think about solvent, a lot of the times as a liquid, and solute could also be a liquid or a solid. But even the air that we are breathing in right now is a solution as well, so because it's an even mixture of all kinds of different types of gases. And in this case, we will say nitrogen is the solvent because nitrogen is the most prevalent out of the different types of gases. And carbon dioxide and oxygen, for example, will be the solute. So here's another picture showing you the difference between a regular mixture, right? Even a salad, that's a mixture, but a solution could be a lemonade, or if you're putting different chemicals together, you can see this even mixture. Can you only mix solid and liquid to form a solution? The answer is no. Why is water a good solvent? So water is really good at dissolving things because water is polar. So it's able to grab onto other types of molecules, kind of pull it around and get it all mixed up. So water is a good solvent because it's polar. What is the suspension? So suspension is an uneven mixture. So for example, this one right here shows you vinegar and oil. They can mix for a little bit, but then over time, the vinegar and the oil will separate. So is sand and water. If you put some sand in water, you could make it almost look like a solution, but after a while, the water and the sand will separate on its own. So you call that a suspension, an uneven mixture that can separate on its own. Factors that make a suspension different from a solution is whether it can separate on its own or not. Do molecules move faster or slower when the temperature of a solution raises? So think about dissolving sugar into water, right? Let's say you're making lemonade. You're putting sugar into this lemonade and you try to dissolve the sugar. It's much easier if you're heating up the solution. The next one we're going to talk about is pH. So pH is something that measures acidity. When you have a solution, you can say that the solution is acidic, or the solution is neutral, or it's basic. So this is a property of the solution. When you think about water, for example, water is what we call a neutral a substance, because water can dissolve into hydroxide ion, OH-, and hydrogen ion, which is H+. When you have an equal amount of OH- and H+, if you have water, you would have equal amount of both, then this substance will be neutral. When you have higher concentration of hydrogen ion, that will make the solution more acidic. And when you have higher concentration of the hydroxide ion, or in reverse, the lower concentration of the hydrogen ion, then you will say this solution is more basic. So pH, it has H in there, so pH actually measures the concentration of hydrogen ions. We represent the pH of a solution from 1 through 14. So when you look at this picture right here, the more you are on this side of the picture, the more acidic the solution is, and the higher the number is, the more basic this solution is. Okay, so in the middle we have 7, which is neutral. When you have a pH 6, for example, it's actually 10 times more acidic than 7, and pH 5 is 100 times more acidic than 7. So the smaller the number gets, the more acidic the solution becomes and the higher concentration of hydrogen ion there is. So I'm going to write this one down for you actually. So we have smaller number, more acidic, higher concentration of hydrogen ion, lower concentration of hydroxide ion. All right, so this is what this means. When you in comparison, if you have a larger number of the pH, it means it's more basic or less acidic, and it has lower concentration of hydrogen ion and higher concentration of hydroxide ion. If I were to ask you a question about a more acidic acid, right? So if I were to have pH 1, pH 5, pH 9, and I said, which one is the most acidic acid? It will be 1, right? Or if I were to say, which one is the least acidic acid, that would be 6, and not 9, because 9 would make it basic, and 6 is actually acidic. So if I say more acidic acid, that would be 6, right? And pH could have decimal as well. You can have 6.3, for example, as the pH value. So let's take a look at the questions. What is the range of numbers on the pH scale? That would be 1 through 14. The range of acidity is 1 through 7, or just smaller than 7, bigger than 1, smaller than 7. The range for basic is bigger than 7, smaller than 14. Neutral will be pH equals to exactly 7. What molecule is neutral? That would be water. If you're comparing pH 5 versus 6, the more basic one will be pH 6, and the more acidic one will be pH 5. If you're comparing pH 8 versus pH 11, the more basic one will be 11, and the more acidic one will be 8. So the higher the number, the more basic it is. If you're comparing 5 and 6, which one has higher concentration of hydrogen ions? That will be pH 5, because it's more acidic. The smaller the number is more acidic, more higher concentration of hydrogen. And then pH 8 versus 11, pH 8, although pH 8 and 11 are both basic, pH 8 would have higher concentration of the hydrogen ion, because in comparison, pH 8 is more acidic of a base compared to pH 11. Moving on to macromolecules. So macromolecules are organic molecules that there are 4 big categories of it. What elements do carbon bond? So carbon is really good at making bonds. Each carbon can form 4 bonds, and carbon can bond with hydrogen, oxygen, and nitrogen, and phosphorus to make up other types of molecules. It can bond with a lot of other things as well, but carbon generally bond with these types of elements to make these macromolecules. Can carbon bond to other carbons? The answer is yes. How many bonds does the carbon make? That would be 4. So carbon has 4 valence electrons. Each one can make 1 bond. What is a macromolecule? So macromolecule is an organic molecule that is quite large compared to other types of molecules. So macro means large, right? So it's a large molecule. What is a monomer? What's a polymer? So when you have a macromolecule, it's often made out of repeating units of something. So when you have a polymer, that is the overall repeated unit of the material, and then if you only have one piece of this repeating unit, then you'll have something called monomer. There are 4 types of macromolecules. So this is a monomer, and a polymer is made up of monomers, right? So then when we could talk about the macromolecules and what are the monomers of each type of macromolecule. What you have here, there are 4 types of macromolecules. There's carbohydrate, lipid, protein, and nucleic acid, and we can look at questions based on each one of them. For carbohydrate, the elements that make up carbohydrate is carbon, hydrogen, and oxygen. The carbohydrate monomer is called monosaccharide. And specifically, when we have 2 monosaccharides together, we call it disaccharide. And when you have a polymer specifically for carbohydrate, it's called polysaccharide. Examples of carbohydrate, we have glucose, cellulose, right? Glucose is what we use directly for cell respiration. Cellulose makes up of cell wall of plants. And then functions, hi. Let's continue recording this video about carbohydrates. There are a few things that we can discuss, right? And we got to the part where we say the examples of carbohydrates are glucose and cellulose. And there are other types, you don't really need to know them. Functions of carbohydrates. So carbohydrate is good for storage of energy, especially a short-term storage of energy. Because usually if you have long-term storage of energy, that would be lipid. And carbohydrate is also used to build cell structure and things like that. Moving on, lipids. What elements make up lipid? That will be CHO as well, carbon, hydrogen, and oxygen. What do we call the two monomers of lipids? That will be glycerol and fatty acid. So when you look at a picture of a lipid, this animation, for example, we have this body part, this head part, and we have the tail part. The tail parts are the fatty acid, and the head part are glycerol. Generally, overall, lipids are nonpolar, right? Nonpolar meaning that they can't interact with water, which is why oil cannot be mixed with water. So the fatty acid part of a lipid is really what makes it really nonpolar. When you look at a cell membrane, the phospholipid has a head and two tails. The head part is hydrophilic or polar, and the tails are hydrophobic and nonpolar. So that's lipid. Examples of lipid, clearly you would have fat, oil, phospholipid, cell membrane, right? This last one is really important because overall, we're talking about biology, we're talking about living things. Fat and oil are really important for insulation. So insulation, long-term energy storage. But I would think the most important of all of them is the phospholipid bilayer of the cell membrane, because every single living thing, every single cell needs the cell membrane. Oh wait, that's examples. So the function of the lipid will be long-term energy storage and build structures as well for cells and organisms' bodies, and for insulating the bodies, right? What is the difference between saturated and unsaturated fat? So the difference is, well, for this one, you actually need to find a picture. So when you look at a saturated fatty acid, this is how it looks like. So a saturated fatty acid has carbon-hydrogen tail, but this hydrocarbon only has single bonds in between, and it's a straight tail. And for unsaturated, it's different because unsaturated would have a double bond somewhere within the carbon hydrogen bonds, and then you form this unsaturated fatty acid. It's unsaturated because it doesn't have as many hydrogen compared to the saturated fatty acid. All right, moving on, which type of fat is liquid at room temperature? So if you have saturated fatty acid, you can squeeze a lot of them really tightly together, then that will be a solid at room temperature. But if you have unsaturated fatty acid, because you have this kicking of the tail, which is called a kink, if you have these saturated acid being packed, they don't pack as tightly together, so then you will have a liquid at room temperature. So let's say butter, for example, will be an unsaturated fatty acid. No, I'm sorry. Will be a saturated fatty acid because they're able to pack all these fatty acids together and form this solid at room temperature. And olive oil, for example, will be unsaturated fatty acid. Moving on, proteins. So elements that make up proteins are carbon, hydrogen, oxygen, and nitrogen, CHON. What do we call the monomers of proteins? Those are called amino acid. And I think you guys are pretty good at this now. What makes different amino acids different? So they have different, what we call R groups. So if you were to look up a picture of an amino acid, this one, for example. So when you look at this picture, you see that they all have a very similar structure. It has the amino group and the carboxyl group. And then the bottom part, or in this picture, they're on the bottom, but really you can rotate it any way, right? This part is what we call the R group. This part can change into all different forms, and this R group is what makes different amino acids different from each other. And sometimes you would have some polar R groups. Sometimes you would have an R group that even has a charge on it, negative charge. For example, you could have positive charge, or it could be nonpolar. So these R groups are able to interact with each other and allow a protein to fold into a shape, a very specific shape that allows the protein to function. So the R groups. Enzymes and catalysts are examples. Well, this is under proteins. So these are proteins. So enzymes are proteins that are specifically in charge of speeding up certain types of chemical reactions. What do we call polymer of proteins? Those will be called polypeptide because in between each type of the amino acids, when you put the amino acids together, the bond in between is called a peptide bond. So a polymer of protein would be called a polypeptide. A polypeptide has to do what to make a functional protein? It has to fold. So this folding process is really important because it's kind of like if you have a paper, right, and you're trying to make a paper airplane. You have to fold this paper airplane to make it fly, right? Same idea with proteins. When you put amino acids together, this is just one long chain. It can't do anything if it's just one long chain. It doesn't have a shape, so it can't have a function either. So what has to happen is that different parts of this one long chain of amino acids has to interact with each other in order to fold into this some sort of shape that works really well, once it's folded correctly. So it's as if you fold a paper airplane correctly, then it will fly, but if you fold it incorrectly, then it wouldn't fly, kind of thing. Functions of proteins. So proteins are in charge of, sometimes they're in charge of transport. So you can remember in the cell membrane, there are proteins involved for molecules to transport in and out of the cell. The proteins can speed up chemical reactions for sure. Proteins can also be in charge of building structures of the cell or the body, right? Protein denature. Protein denature is the unfolding of protein under really high temperature. So if you have really, really high temperature, that's when proteins, they will fall apart. The folding doesn't work anymore. Moving on to nucleic acid. So nucleic acid has CHOMP, so carbon, hydrogen, oxygen, nitrogen, and phosphorus. What do we call the monomers of nucleic acid? Those are called nucleotides. Examples of nucleic acids, the ones that you have to know is DNA and RNA. Nucleotides all have three parts. They have nitrogenous base, it has a sugar, and it has phosphate groups. Functions of nucleic acid: storage for genetic information. That's what DNA is for. RNA is to help make proteins. And then you have ATP is actually another example of nucleic acid, so it also works for energy. I'm talking about energy and chemical reactions. You need to understand how a reaction happens, right? We have the reactants, we have the products, and you've learned that multiple times, and I think you're really good at it already. But with reactions, there are two different big categories. You have spontaneous reactions versus non-spontaneous reactions. How this works is that if you think about it, right, if you want to burn something, this is a pretty easy process. So this will be an energy releasing reaction. So this is a spontaneous reaction as well. If you think about iron getting rusty, this is a spontaneous reaction. It can just happen while the iron is sitting in the air, right? If you were to say, let's say you're trying to turn water into hydrogen gas and oxygen gas. This is not a spontaneous reaction. This doesn't happen on a daily basis, right? If you don't actually put in some sort of extra work into this process, this reaction is not going to happen. That's what we call non-spontaneous reaction. So when we talk about spontaneous reaction, you could look at graphs like this, right? You need to understand what this graph is telling you. We have the course of the reaction on the x-axis, or time of the reaction, and the y-axis is the energy level. What you see is that we have these reactants that start at a certain energy level, and then the energy level has to go up. So there has to be an input of energy of some sort, and then the energy level goes down, and eventually you reach the bottom part, which is our products. So what you see here is that the reactants started right here, and products ended right here. Although you have this bump that you have to go through, overall you're lowering the energy level, right? So if you're lowering the energy, all you have to do is release the energy, and this process will be a spontaneous reaction because releasing energy is easy. So spontaneous reaction, my definition would be, it's a reaction that can happen on its own without the input of energy. All right, moving on to what type of reactions are usually spontaneous. That will be energy releasing chemical reactions. The non-spontaneous reaction will be the energy absorbing chemical reactions. For example, if you look at this picture right here, we start right here with the reactants. It goes through this bump, and then the products land it right here. So overall, you have to absorb more energy. If you're trying to absorb energy, that energy has to come from somewhere, right? So that energy has to come from a real energy input. So this, you have to find this energy somewhere, right? So this will be a non-spontaneous reaction because this reaction cannot happen on its own. The next one is called activation energy. So activation energy is the amount of energy it needs, the amount of energy a chemical reaction needs to start the process of the chemical reaction, right? So for example, whether you have an energy absorbing reaction or an energy releasing reaction, you all have to go through this bump. It's as if you were trying to go to the other side of the hill, you have to go up the hill and then go down the hill a little bit, right? Even if you're going down the hill to a place you end up much lower than you started, you still have to go over the hill. Is this reaction spontaneous? Or you can think about this. If you're shooting a boat, right? In order for the boat to shoot, you have to pull I don't know what it's called. You have to pull the boat, right? You have to pull it first before the arrow can shoot. So the arrow can shoot really far, but you have to have this initial input of this energy, right? To allow the shooting of the arrow. So that's what activation energy is for. So when you look at this question, is this reaction a spontaneous or non-spontaneous? This will be a non-spontaneous reaction because it's an energy absorbing reaction, because the product energy level is higher than the reactant's energy level. Is this reaction spontaneous or not? This will be an energy releasing reaction, so it's a spontaneous reaction because the product's energy level is lower than the reactant's. The last part that we're going to talk about is called enzymes. So enzymes are proteins that speed up a chemical reaction. So how does an enzyme speed up a chemical reaction? It speeds it up by lowering the activation energy. So normally you have to go through this energy bump, right? This energy bump, this activation energy can be any amount of energy. But for a specific reaction, sometimes if you use an enzyme, you'll make this reaction happen more easily by making this bump lower, right? So the requirement to start this reaction is not as much. Then this reaction is easier to happen, which is why the enzyme can speed up the chemical reaction. What do we mean by enzymes are specific? So enzymes are specific because enzymes all are proteins, so they all have its own shape. It's like a lock and a key. This lock, right, or this key can only open certain locks. And this key has certain shapes, but you only use one key to open one lock. If you're opening another door, then you need generally using a different key, right? But this key can be reused. So enzymes, once it's done its job, it can be reused again. How does temperature affect enzyme activity? Because as temperature gets higher, molecules move faster. So no matter what kind of reaction it is, the higher the temperature, the faster the activity of the faster the chemical reaction can happen. When you're involved in an enzyme, generally temperature will increase the enzyme activity unless the temperature is too high. So what happens at really high temperature, again, is that a protein can fall apart, right? If a protein falls apart, it can no longer work. How does a pH affect enzyme activity? Generally, enzymes have an optimal pH, meaning that at this pH range, the enzyme can speed up the reaction the fastest. So if you have a lower pH or much higher pH, the result is this enzyme activity is not going to be as high. Now, when we're talking about, there's a word called optimal temperature or pH. So if you look at this picture, you will see that there's this particular enzyme and the activity is, well, let me do this one. The rate of the reaction is highest at this very top, right? Whenever you see this type of picture, you want to look at the very top or the highest bump of the line, and then the corresponding x-axis will be the optimal temperature or pH. So the optimal temperature for this reaction will be about 37 degrees. Okay, and different types of enzymes would have different optimal temperature or pH, right? So for example, if you look at this one, we have the optimal temperature of enzyme A will be about 36.8 degrees, and then the optimal pH will be maybe 6.8. And then enzyme B, however, in comparison, because it's a complete different enzyme, has different activities. So for enzyme B, it has an optimal temperature of about 37.9, and the optimal pH is about 7.4.